So I'm reading now from your lecture handout booklet. It turns out you already had some description of organogenesis right there at your fingertips all along. Mostly this doesn't add new information to what you've already been writing about from the previous organogenesis video, but there's a few new pieces in here you might add to your description of organogenesis. So here we are looking down at my drawings from the slides. And in the first two drawings on the left, we're looking down on the blastoderm, which is the disc with the developing embryo, from above. Development proceeds in the chicken as it does in all vertebrates from anterior to posterior in waves. By the 18th hour in the chicken embryo, gastrulation's nearly finished. Organogenesis is well underway by the 24th hour. This view shows the neural tube beginning to roll up in the embryo's head, and neural folds are present. Moving down the embryo during this stage, the mid-body, the neural plate's still present and not yet forming a neural fold or becoming a tube. Further down the embryo, near the beginning of the primitive streak, the neural plate hasn't even formed yet. Because development in the anterior of the vertebrate has progressed further, moving posterior in the embryo is like moving back in time to an earlier stage of development. By 24 hours, the primordial nervous system is the most prominent of the body organ rudiments. These will form the brain and the spinal cord. Dorsal ectoderm thickens into the neural plate. A groove develops from anterior to posterior in the plate. And the sides fold up to form the neural folds and then the neural tube. In the same region of the developing embryo, bilateral pairs of somites are pinching in mesoderm just lateral to either side of the notochord, which is beneath the neural tube. Somites are the precursors of the vertebrae and the body wall muscle. Below we can see a cross-section through a developing vertebrate that shows the position of the developing nervous system from another angle. After the 24-hour time period, the most anterior parts of the head containing the foregut and the brain are no longer touching the blastoderm because of the uplifting of the head caused by the formation of the head fold. Endoderm forms the lining of the gut tract and associated organs like the liver and the pancreas. In the head fold, the primordium of the foregut, which is called the anterior intestinal portal, is completely isolated from the yolk, whereas the gut opens to the yolk more anteriorly. Just posterior to the portal, the heart will form from a thickened region of mesoderm associated with the endodermal layer just posterior to itself. One thing that chordates have and share with some other bilaterian animals is a column. The column is a fluid-filled cavity that lets the viscera stay free from the body cavity wall. Here the guts slosh around in a fluid bath. A true column is always completely lined with mesoderm derivatives. And this layer, mesoderm derived, begins to form at 24 hours in the chick. The layer splits and the dorsal side, or somatic mesoderm, associates with the ectoderm and forms body wall, muscle, bone, and connective tissue. On the ventral side of the split, the mesoderm forms the heart. The column is much larger by the 33-hour stage, which is the figure above to the left, and most of it is beside the developing embryo rather than in it. We've been seeing this in our candlings. This extra embryonic region beside the embryo is the yolk sac, okay? And these regions will form the membrane, including the amnion in the next stage of development that will come to surround the embryo completely. Once the vascular tissue is in place to feed and allow the developing embryo to expel wastes, this will occur. By this stage and into the 48-hour stage, the notochord is still proceeding to develop posteriorly as the neural plate and then the neural tube finish forming dorsal to the notochord. the neural tube has formed into a hollow tube and becomes subdivided into a forebrain, or telencephalon, with lateral optic vesicles, a midbrain, diencephalon, which will grow outwards towards those optic vesicles, and a hindbrain, mesencephalon. In the 48 and 72-hour stages of the neural tube has formed a structure that resembles the brain of the juvenile shark we examined in class. Actually, we didn't do that this year because of the pandemic. In this unique case, where von Baer is correct, and ontogeny recapitulates phylogeny, von Baer's famous quote, we can show that the shark brain retains many ancestral characteristics. Because the shark's brain looks much like the chick's brain in early development, with a forebrain, a midbrain, and a hindbrain, we can say that it resembles the brain of the ancestor to all chordates. The chicken brain will not change much in global architecture as the chick develops. This is unlike a mammalian brain, which includes a processing center with two hemispheres called the neocortex, or new cortex. See the cross-section from the lab on the next page. At the 33-hour stage in the chick, the heart is a rather simple tube. Blood enters the heart tube posteriorly from the bilateral vitelline veins. These veins arise on the yolk sac, where the heart receives oxygen and nutrients, and meet on the midline to form the sinus venosus of the heart. This chamber leads to the atrium, which leads to the ventricle, where the heart tube curves to the left. The outflow part of the heart, the conus arteriosus, swings back into the midline again and gives rise to the paired ventral aorta, located ventral to the foregut. It's important to note that much of the vascular tissue is still external to the embryo, even further into organogenesis. The 48-hour chick bends into the fetal position, causing the anterior part of the head to face posterior around the level of the heart. This is the position the chick will remain in until it chips itself free, unzipping from the egg with its egg tooth. Notice the eye. The eyeball forms from the forebrain wall, or the ectoderm, actually external overlying the forebrain wall, okay, and will grow a lens that will attach to growing diencephalon from the brain. And while the lens will form from the overlying ectoderm, the underlying parts of the eye will form from this protuberance of diencephalon. Because of the new position of the embryo bent in the fetal position and turned sideways, the mouth will actually form at the same level as the eye. Here we have definitions of important terms then on this page that we've gone over during our unit of development. So here we're looking in the head region of the embryo, and we see the expression of OTX1 and OTX2 will cause these lateral protuberances to grow in the diencephalon wall near the mesencephalon. These protuberances are going to grow towards the overlying ectoderm. The overlying ectoderm will express PAX2 and PAX6 and form the lens of the eye, and all of this together will form the developing eye placode. So here we're looking at vertebrate limb development, and we're going to understand actually that the hind limb develops similarly as far as Hox gene expression is concerned to the forelimb on the proximal, which is closest to the body, to distal, which is the end of the limb, fingertips or toes, axis. Now, we review what we studied earlier this year, and we see that at the anterior expression boundary of Hox6C, this is step 1, establishment of the forelimb bud, that is where the apical ectodermal ridge or rudimentary limb bud forms. The apical ectodermal ridge, or AER, is a tissue of cells. These cells will then express FGF4. That's fibroblast growth factor 4. Now, the cells on the posterior side of this limb bud, called the ZPA cells, or zone of polarizing activity, will receive this FGF4 signal, and they will express the gene sonic hedgehog. This will cause the posterior anterior axis of the developing forelimb bud to form. Now, if we look at number 3, we see that the development of the posterior to anterior axis of the stylopod, that's the part of the arm from the shoulder to the elbow, and the zeugopod, that's the part of the arm that is elbow to the wrist, and the autopod, that is the hand of whatever vertebrate it is controlled by Hox gene expression. So sonic hedgehog sets up the initial posterior-anterior boundary, and then we see that Hox10 is expressed on the posterior side as the stylopod grows out, and Hox9 on the anterior side. And then we see Hox13 is posterior in the zeugopod, expressing all Hox genes to Hox10, which is anterior in the outgrowing zeugopod. But then finally, for the autopod, the phase 3 enhancer is expressed, and it flips the anterior-posterior regime of Hox gene expression, so that now the most anterior part of the hand expresses Hox13, then Hox12, Hox11, and Hox10, heading towards the posterior or pinky side of the developing vertebrate forelimb.